IB Biology Topic 3.2 Chromosomes Point 4. In a eukaryote species, there are different chromosomes that carry different genes. In this video, we'll be looking at why and when we see different chromosomes under the microscope. Our first point is that eukaryote chromosomes are too narrow to see during interphase, although they become fatter and shorter during mitosis and meiosis. This is called supercoiling. Supercoiling. Once supercoiled, we can see these chromosomes under a microscope if we use a stain. And so this next point is, these are visible if stains that bind to either DNA or protein are used. The chromosomes also look like they double during the first stage of meiosis. So during the first stage of mitosis, the chromosomes can be seen to double. And this effect is seen because there are two chromatids with identical DNA that are produced during replication. When we look at eukaryote chromosomes during mitosis, during mitosis, when we look at these chromosomes, we can see that there are differences between the chromosomes, and so we say that there are different types. Different types that may be seen, and they differ by length as well as by the position of the centromere. Now, if you don't remember what the centromere is, when we look at a chromosome, this little piece that connects the two chromatids is the centromere. So here we have the chromosome. Each one of these is called a chromatid. So we have one on the right side and we have one on the left side, and this thing in the middle is the centromere. The centromere can be almost anywhere along the two chromatids, so it can connect the chromatids more at the top, in the center, in the bottom, and so on. And depending on where we see the centromere, we can identify the different types of chromosomes. And this, of course, is as well as by looking at their length. And when we look at eukaryote DNA under a microscope, we see that a eukaryote must have at least 2 chromosomes. And furthermore, in humans, there are 23 chromosomes. Now, this 23 is 23 unique chromosomes, because in reality we have 23 pairs, so we have 23 times 2, which is 46. We will also be looking at genes in chromosomes point 4, and so the first point is that every gene has a specific location on a chromosome, and this specific location is called the locus. And so we also say that for each chromosome, when taking into account the genes, is a unique linear arrangement of genes. So this is a sequence that may have well over 1,000 genes. So I'll just write 1,000 plus genes. And one more point here is that because genes on a chromosome have a specific location and order. In this case, the location and the order are the same because a gene takes its position along the chromosome, and in this case, this will also be the same as its order when in comparison to the genes in front and behind it on the chromosome. And so because genes on a chromosome have a specific location and order, gene swapping may occur during meiosis. And also, to clarify gene swapping, as a separate point, I'll write that gene swapping is the same as chromosomal crossover. And this is also called crossing over. And this is the exchange of genetic information between homologous chromosomes. And this exchange of genetic information, we call this genetic recombination, and this allows for most of the genetic variants that we see in life. And the reason that this can happen is that when we have homologous chromosomes, so this will be a paternal and a maternal chromosome, meaning from the father and from the mother. In this example, we're going to be speaking about humans. So we have two chromosomes, paternal and maternal, from the same species, and they're homologous, meaning that this is a set of chromosomes that pair up with each other during meiosis. So this all happens during meiosis, and this chromosomal crossover happens specifically during prophase 1 of meiosis. Now, as I mentioned earlier, there's a specific order and location for every gene on a chromosome. So when we have two homologous chromosomes, the paternal and maternal, and they pair up together, we can experience gene swapping because the same gene that you're receiving from your father and from your mother is going to be on the same in the same locus. So it's going to be in the same location and the same order on both of the chromosomes. And during meiosis, we experience this recombination because, as just from this name, crossing over, some of the genetic material can cross over between the two chromosomes. And from this, we get the mixing of genes called genetic recombination. And to help visualize this, I've drawn two chromosomes, and these are homologous chromosomes. So on this paternal and this maternal chromosome, you'll have corresponding genes in the same order. And so, for example, let's say this gene and this gene, they have the same locus because they correspond to each other. And during meiosis, they can become exchanged, swapped. And here we see that one crosses over, takes the position of the other. And to reiterate once more, this is because they have the same locus. And this is why it's so important that every gene has a specific locus, and thanks to our biology, the genes will automatically take this position and no other.